Okay, let's begin. So, I believe we stopped here with lateral inhibition. All right. But I know you have a question, so let's take that first. Okay.
I could, when I went home to try to explain it to myself, I couldn't. So I just want to know why does, for the arm cell, why does it release more glutamate? Is it because the light is shining on it?
So, let me... You're referring to this?
Yes.
This one, right? Okay. That's the one? Okay, so here what we have is an example of the pathway from the photoreceptor. Here we have a cone, all the way to the ganglia, taking the information to the brain. Okay? This happens to be an on-center cell, which means that, well, there's both an on-center and off-center bipolar cell. So here, we're looking at what happens to the ganglion cell in terms of, is it going to fire an action potential? Is it not going to fire an action potential? Given that the photoreceptor, the beginning of the pathway, is exposed to light or not exposed to light. So here, the center is exposed to light. Okay? So the cone gets exposed to light. As a result of that, now remember that the photoreceptor typically, in the absence of light or in darkness when there's no stimulation, it's typically continuously releasing glutamate. That is its natural state, continuously releasing glutamate. As a result of exposure to the light, you actually get a reduction in the glutamate released. You get hyperpolarization, which is kind of like the opposite of what we talked about in chapter 4. So that's a key thing in understanding this, is that the activation of this cone actually leads to hyperpolarization. Whenever you have hyperpolarization, you have less release of the neurotransmitter. And the neurotransmitter here in question is the glutamate. Are you with me so far?
Yes.
Does that make sense?
Yes.
Okay. So less glutamate. Now, you have what's called an on-center bipolar cell, and you have an off-center bipolar cell. These two types of bipolar cells react to glutamate in opposite manners. So the on-center bipolar cell is typically inhibited by glutamate. Meaning that for this on-center bipolar cell to be activated, you actually need to lessen or reduce the glutamate being released by the cone or by the photoreceptor, which happens when you have the cone exposed to light. Okay? The off-center bipolar cell is typically excited by glutamate. Now, excitation means that this particular bipolar will release more glutamate, which means that it will be inhibited by a reduction of glutamate. This reduction of glutamate happens when the cone here, or the photoreceptor, is activated by light. Okay? So the on-center cell gets activated because you have less glutamate being released, and it is inhibited by glutamate, so you take away that inhibition by releasing less glutamate, allowing it or taking your leg off the brake, so then it's free, the on-center cell, to release more and more glutamate on the next cell, which is the bipolar cell. And the bipolar cell here is the only one out of all these cells that can actually fire action potentials. So in response to the increasing glutamate, well, it also increases its firing rate. For the off-center cell, if you have the photoreceptor being activated by light, right, you're going to have less glutamate being released. It's usually excited by glutamate, but now there's less glutamate, so it's going to release less glutamate. So the off-center bipolar cell will end up releasing less glutamate on the bipolar cell, on the ganglion cell. Less glutamate on the ganglion cell. Because the ganglion cell, it doesn't matter if it's an on-center or off-center ganglion cell, it's always excited by glutamate. And the more glutamate, the more excitation. And what that means is an increase in the action potentials. Therefore, it releases or decreases firing rates. Does that make sense? Where do things fall apart for you? And probably for many people in the class.
I think it's the on and off that are confusing me. So for the off center, it releases less glutamate because the light is shining on it.
Yes.
It wants it to be turned off. So
Yes.
it will release more.
Yes, that's why it's called off center. Think of off center as off light and turned on.
Okay.
Think of on center as on light and turned on.
Okay.
Okay, that sounded weird for some reason. Especially at 8 o'clock in the morning. Okay, does that make sense?
Yes.
Okay. Any other questions before we go on? Okay. So then with that, I think we stop here with lateral inhibition. Okay. Making kind of the point I started with the visual system is that really your brain represents or presents you with perception not necessarily of reality out there, physical reality out there. Lateral inhibition has to do with a process in which sensory receptors, okay, so here we're talking about the visual sensory receptors, cells inhibit information from neighboring receptors. So, and this is what helps you see contrast between edges. So I don't know, I don't know if you've ever noticed, but like if you see a black sign and it has white writing in the middle, you find that the white lighting is kind of like shining, or the edges of the white words, it's kind of like shining outside. Next time, notice that's lateral inhibition. Okay, so it's basically the bipolar cell. If a bipolar cell is activated by a photoreceptor, given that slide that we discussed, then what happens is it actually inhibits its neighboring ganglion cell. So this leads to our ability to see contrast or to see edges in corners really well. Okay. So then when the ganglion cell is inhibited, it's going to carry less information about that part of the receptive field to the brain. So again, visual experience is not actually just a simple reporting of the physical world out there. This is something that the brain creates for you that makes you more adaptive out there in nature to your environment. Because seeing borders, seeing corners is really important. Because think about if you're going on a cliff, and if you are not that good with seeing borders or corners or edges, then you might just fall. Okay. All right. So here you have the receptor cells depicted in the purple, and then you have the ganglion cells in, I guess, yellow or mustard. And what you have here is if you activate the ganglion cell, or an activation of the ganglion cell, will lead to the decrease in firing of the neighboring ganglion cell. Okay? Because remember that the ganglion cells talk to each other. There are lateral connections. So this leads to this. Do you notice, if you actually cover this, do you notice that the edge of the line is brighter on one side? Okay. Which color is brighter here? Okay. Now actually, if you have your book, does anyone have their book? Now I want you to put your finger and cover that white line and tell me what do you see. Yes. They're actually exactly the same color. The top and the bottom. Then the top and the bottom is actually exactly the same color. I love doing visual illusions like this when I have couples fighting and I'm trying to coach them. And I'm like, okay, which one of you is brighter? Would you put money on it? Yes, this one is brighter. I swear it is. I can put my savings on it. And I'm like, oh yeah? Okay, look, they're actually the same color. There's no such thing out there as reality. Your brain creates reality as you go on in life.
That's scary. What are you doing?
Really, all of you guys, I'm hypnotizing all of you guys, but you don't know. Okay, but actually they're both exactly the same color. All of the squares, all of them are the same color.
That's pretty cool.
Okay? Again, making the point, your brain is creating perception. Really, so there's sensation, which is the actual physical reality out there, and then there's perception, what your brain wants to make out of that reality. Okay, neurons of the visual cortex have very different receptor fields. Okay? So remember we talked about the idea of receptor field before? So neurons for the ganglia are going to have receptor field, but also neurons in the cortex. That's all that information is going to go to the brain. At every single layer, at every single level of processing, each neuron is going to have its own receptive field that's organized in a topographic map. So remember I said that most of the ganglion cell axons, you're going to have the optic nerve, and then they're going to crisscross at the chiasm, not all of them, and then they're going to go where? Then the becomes the optic nerve, and where does it go? Or sorry, the optic tract, and where does it go? To the lateral geniculate nucleus, exactly, the LGN, which is located where? In the thalamus. Okay? And the thalamus is a collection of nuclei. Each nucleus is going to process different kinds of information. Does anyone remember what nucleus means? And I don't mean the nucleus that's located in the soma that houses your DNA and genetic material. I don't mean that one. That is a correct definition, but that's not the one I mean here.
Isn't it the same thing as ganglia, but in the brain?
Exactly. It's the same thing as ganglia, but in the brain, which is a collection of cell bodies and dendrites. So basically nothing white. No myelinated axons. Okay? So, and each auditory cortex is going to have its own dedicated nucleus in the thalamus that processes this for all the sensory modality, except for olfaction or your sense of smell. Okay, so the neurons, or the LGN visual neurons, are going to have receptive fields. Remember when we talked about the retina, the receptive fields were concentric. Circle with a surrounding circle. Yes? Remember that? Now the receptive field for the LGN or the LGN neurons are elongated, oval kind of like shape. So the LGN is going to send its axons to the visual cortex, the primary visual cortex, which we said is V1, or the striate cortex. They're exactly synonymous. And then in the striate cortex, these cells are going to process certain information. We're going to talk about that later. That has to be elongated, or elongated stimuli. And we'll talk about what that means when we talk about the cortex. Okay, you have then in the cortex what's called, in V1, you have what's called simple cortical cells and complex cortical cells. Simple cortical cells see bars or are edge detectors. Sometimes in books they refer to it as edge detectors. And that is because they respond to an edge or bar of a particular orientation and location. So by orientation, I mean how tilted the line is. Is it 45 degrees? Is it 90 degrees? Is it horizontal? Is it vertical? Is it 270 degrees? That's what I mean by an orientation. So it has very specific neurons that will respond to all the different orientations 360 degrees around. Okay? So meaning that if I'm measuring from a specific cell in the V1, if I have a line that's oriented, let's say that this is a cell that gets turned on by horizontal line, then the opposite of that would be a vertical line. Then if I present a horizontal line, you're going to hear a lot of action potentials. If I present a vertical line, you're going to hear almost zero action potentials because it's turned off then by vertical lines. Okay? So the maximal stimulation for that neuron is horizontal line. You know, you have something maybe 1 degree, 2 degrees off that, a little bit off that. It's going to get so stimulated, although that stimulation is going to weaken and weaken the further you go away from horizontal, and certainly gets to zero by the time you get to vertical. Does that make sense about the word orientation and what that means? Okay. And then you have complex cortical cells. They're called complex because they process more complex information than simple cells. So they also respond to a moving bar, okay, with a particular orientation, but this orientation, it can be located anywhere in the visual field. So a lot of this research has been done on kittens and cats, and actually this information was discovered by accident. And if we have time, I'll show you the video of like how they actually moved the line, and you actually hear the action potentials. Yeah, hopefully I'll have time to show that. Okay, so basically they have an electrode, a microelectrode inside the visual cortex, V1. They show a moving line like this with a certain orientation, and then they look at the number of spikes or action potentials. If that cell is turned on by that orientation, then it will fire action potential. The decrease in the number of action potential means that the cell is losing interest in that orientation. So that's how they figure out the orientation, or which orientation best matches the different neurons in the V1. Okay, so here are examples of receptive fields. We already said that before it gets into the brain, we know that they have concentric receptive fields, a circle and another circle. So in the thalamus or the lateral geniculate nucleus, you also have concentric field, receptive fields, a circle surrounded by a circle. And then you have all of the stuff we talked about. If the center is illuminated, the cell might be turned on, which is an on-center, off-center surround. What I mean by surround, it means the circle, the outside circle. Okay? And then you also have off-center, on-surround, which is when the surround is illuminated. So you can play with all those four combinations. Okay? So the same idea there remains the same for the lateral geniculate nucleus. Although the receptive fields are going to be a little bit bigger than the receptive fields we talked about earlier. Okay, then you move into V1, the cortex, and you have the simple and complex cortical cells. The simple cortical cell is going to be sensitive to the orientation of a line. So now you're talking about receptive fields that are elongated as opposed to circles with light in the middle or the center and off surround. So a little bit different and actually more complicated. At this level, we're already now getting to a level that you can appreciate, like an actual line. Okay, so a simple cortical cell sensitive to this orientation. So do you see how the horizontal line, what do you see? Do you think which line orientation is this cell most turned on by? Vertical lines. But do you see when it's a little bit off vertical, you still get a little bit of action potentials? When it's horizontal, you almost get zero. Because it's the orthogonal or the opposite to the vertical. Does that make sense there? Okay. Now with complex cells, so remember here that you're sensitive to orientation and location of that line. If complex cells, you add one more dimension. You have all of the above, but you also add a dimension of movement. Which way is the line moving? So when the line is moving this way, if the line is horizontally oriented and moving this way, you get action potentials. If the line is horizontal, that does not suffice on its own. If it's moving in the opposite direction, you get a reduction in action potentials. Does that make sense? Okay. If the line is vertical, it doesn't matter if it's moving in the right way or not. It's not going to be turned on by vertical lines. Okay, that's why it's called complex cells. So all of the things that turn on the simple cells are also going to be the same. So orientation, location are still important. Well, not so much location because you're moving, but also you're adding the dimension of movement. Which direction is the line moving? Some of the cells are going to be more turned on by horizontal, moving downward, while others will be turned on by horizontal, moving upwards. Is the distinction between simple and complex cells clear? Okay, so visual cells in the LGN have concentric receptive fields, which is the same as all of the other retinal receptors we talked about. Okay, it's as though the LGN nucleus is kind of a part of that. although the receptive fields there get bigger. And then the visual cells in the cortex, they are going to respond to more complicated things. And here you're already starting to appreciate things that you can actually describe, like in the environment, lines, movements. So now we're getting to a level where, okay, that matches our visual experience. Okay. So let's see how that builds up in terms of the receptive field. So the different circles are going to be receptive fields. Okay. The concentric circles are going to be that center and then surround, with the inner circle being the center and the outer circle being the surround. Yes?
And is that the on and off, does that also help us see detail? Is that in this kind of, no?
Okay, yes, it's obviously going to help, yeah, it's definitely going to help you see detail. But I think also the detail that I really mean when I say about the cone sees detail are the ones that have to do with color, your ability to see color. This is not for your ability to see color. We're going to talk about how you see color in a little bit.
So this is more the edge thing, or like the ability to see edges, or no?
This is definitely yes, the ability to see lines, the movement of lines. But then don't forget that you build on these lines, build on to see something more complicated.
Right.
And then so V1 is going to be maybe the lines, the movement of the lines, but then V2 is going to be a little more complicated and more complicated until you appreciate an actual triangle or an actual figure.
I was just trying to figure out why the on and off, like why that's helpful.
Okay, because what you're trying...
Complicated.
No.
Let it go.
No. Okay, so when you were talking about, first of all, like, I want to say, like, everything that's going on in your brain and your body, you have no idea what's going on. You're basically, like, there's a lot of things that are going on, you don't know, okay? So the rudimentary stuff that's happening at the cones, at the rods, at the bipolar cells, the ganglion cells, these are things that you are not going to be able to do one-to-one in terms of what you see in the environment. Do you understand?
Yeah.
So we're going to be building on layer by layer by layer. Even though the previous layers you can't really appreciate in terms of your perception out there in the environment, they will lead to something that you can appreciate. Does that make sense? Okay? All right? Okay, so, or do you just want me to shut up so I'm not more complicated? Okay. Okay, so it's the latter one. All right. Okay, so here's the concentric receptive fields. Okay, that it's kind of like like, kind of like the iceberg. Most of it is buried deep, and then you also just see the top part. It's kind of like that. A lot of the processing is you're not aware of, and then the manifestation, the very end of the processing, then that might be you actually experience. Okay, kind of like you have no idea what enzymes are being released in your body right now, but you know, for example, that they're going to affect your behavior. You have no idea what hormones right now are being released, but you know that, certain hormones might be released and make you feel angry or irritated. But you know that is related. It's kind of the same thing. Okay. So here are 3 receptive fields for 3 different retinal neurons. Okay? To be more specific, for 3 on-center retinal ganglion neurons. Okay? Meaning that these are ganglion neurons that follow the path of remember the bipolar, the on-center bipolar cell that's turned on by light. Okay? That pathway, the one that was on the left. Okay? So turning on light or shining light on the center, which is happening here, that rectangle means there's light being shined on the center of these 3 receptive fields of the ganglion cell. So maximal excitation there because you have the centers being activated. Now you have each of the cell now is going to talk to the next level, or the LGN. So the 3 receptive fields are going to build onto each other. So you're going to have one V1 cell speaking to multiple LGN cells. Right? So it's not going to be one LGN cell speaking to one V1 cell. It's going to be multiple LGN cells, multiple receptive fields building up activating one V1 cell. Okay? Does that make sense? So in a sense, so look at this one here. All the way on the right, you have on the bottom a simple cortical cell. A simple cortical cell is going to be activated or is going to fire action potential when you have a line that's oriented in a specific way. Okay? And it doesn't matter its location because that will be important. It doesn't matter its direct or its movement because that's the complex cells. So right now we're not worried about movement. All we're worried about is the orientation of that line. It doesn't matter where it's located. So here, look at these three neighboring receptive cells where the center is activated, forming, these centers form a line that's what, about 30 degrees tilted? Something like that? Yeah? Okay? And then this simple cell gets information from all three LGN cells that together form this line. And because this is a cell that's activated by a line that's oriented this way, it fires an action potential. It's activated. Does that make sense? I don't know if the silence means I'm starting to speak Greek or that you completely understand. Yes?
So when the lines are in the same shape, the action potential will be, or in the same degree, the action potential will be released?
So when the centers of these neighboring receptive fields for the ganglion cells, and therefore then in the LGN, when they are illuminated in a certain way that when you connect them they make an orientation, like this, then these three receptive fields, now in real life it's maybe more than three, are going to then activate the next simple cortical cell in V1. And it happens to be here that this cell is turned on by a line that is oriented this way, what looks like, no, actually more than 30 degrees. Okay? In other words, what I'm trying to say is that V1 cells are going to be needing more complex information from the LGN. So it needs information from receptive fields or about the receptive field from the LGN for more than just one cell. Because to make a line, you need many different receptive fields to know what the direction of that line is. Okay? Okay, why don't you look at it for a second and see where things fall apart.
So what's the one on the left? That's the ganglion to the
This is an on-center retinal ganglion cell.
Right, and then where's it going?
It's going to the LGN.
Okay.
And then from the LGN, it goes to V1. Okay? So here you're combining receptive fields to make a more complicated perception. Okay. So now we have the simple cortical cell responding to the orientation of the line. Now, you have this simple cortical cell activated by these three receptive fields. You have this one activated by the middle three receptive fields. You have the most right one activated by these three receptive fields. These three different simple cortical cells activation that then feeds to one complex cell, telling it that this line is moving in this direction. And if it's the complex cell that's going to be turned on by a line that's oriented, I don't know if this is 45 degrees, whatever it is, and moving in this direction to the right, then it's also going to fire action potentials. Okay? Yes?
Just to clarify, so the simple and the complex cortical cells, they're only activated in the V1, right? Not the LGN?
Simple and complex, they're only cortical cells. They're only cortical cells. Okay? So you should be able to go from a simple receptive field in the ganglia all the way to a line oriented in a certain way, moving in a certain direction in the complex cortical cell. And you should be able to understand from this how the combination of receptive fields or retinal receptive fields leads to all the way in the cortex to receptive fields that allow you to see moving lines at a certain orientation. Take a second, see if you can answer that question looking at this. No, answer it in your head, not all of you guys at once. Yes.
The LGN neurons, do they connect with both simple and complex cortical cells, or do they go to simple and then the simple go to complex?
Yes.
Both?
No. They're going to go vertically. Yes. It's a hierarchy.
So LGN goes to simple, goes to complex.
Yes. And then so at each level, the neuron is receiving information from more than one to make a more complex message. So you see here that the simple cortical cell is getting information from these three receptive fields.
And then
the complex one is getting information from 9 receptive fields to know about the orientation and the movement of the line. Yes.
I think I'm very confused. I'm like,
Confused? How can you be confused? This is like so clear.
well, yeah. I'm getting, I guess, a little thrown off.
You know, I'm like extremely sarcastic.
Yeah, I know.
understand why you're confused. Okay.
Of the direction of like what's happening. Like you're saying, like the complex cell, it does not go to the LG. I'm so confused. It goes to the LGN.
So let's think. Okay, let's zoom out a little bit. Forget about receptive field. Let's zoom out a little bit. Okay. You have the light. That's the beginning of the pathway. Okay. So write it in your notes. Light. And then going to a photoreceptor. So you have a line or an arrow going from light to photoreceptor. And then underneath photoreceptor, right, that could be cones or rods. Right? Okay? And then from the cones or rods or the photoreceptors, you have then information goes, so an arrow to, bipolar cells. Underneath the bipolar cells, you should write on center and off center. So you know there's two different types. And if I were you, I would write on center minus, meaning it's inhibited by glutamate. But in my head, that makes sense. You write it in whatever way that makes sense to you. And next to off center, I would write plus, meaning it's excited by glutamate. But write it in whatever way that makes sense to you. Okay? Then going from the two different bipolar cells to, what's the next step? Ganglion cells. Now, the axons of the ganglion cells are going to make your optic tract. They're going to crisscross, they make the optic nerve. So from there, you don't have to write that down. Then from there, you go to an arrow to, from the ganglion cells, an arrow that goes to, what's the next area? Lateral geniculate nucleus in the thalamus. You got that one right? Good. LGN. Then from LGN, it goes to V1. But I'm going to be more specific. Okay? It goes to V1 simple cells. And then another arrow goes to V1 complex cells. Okay? Each arrow is going to bring the information to be more perceptually more complex. Each layer adds a little bit more complexity. Okay?
So you can have a V1.
Huh?
You can have V1 and a V1C or V1-3.
So right now we're in V1. We have not talked about
Okay.
yet. We're just in V1 and we're stuck. Okay? All right. Does that make sense? Yes.
So from the LGN, it can go to, oh no, it goes from simple to complex.
Yeah. So from the LGN, it's not. It can go from the LGN. It goes to V1. And then but it goes to simple and then simple to complex.
I thought it was either simple.
No. In order to appreciate, you're building on. Every stage, you're building on information. You have one receptive field. Okay, and then you have 3 neighboring receptive fields activated, forming a line. The orientation of that line is important to simple, and that's all it cares about. But in order for the complex V1 cells to appreciate the movement, it has to get information about the orientation, which must come from simple cortical cells, for multiple simple cortical cells that are next to each other, to tell it which way the line is moving. Okay? Yes.
So in this picture, the 3 there are the simple cells. So the ones before it, the receptor field of those 9 is the LGN?
Yes. Which comes from the ganglion cells.
Thank you.
You're welcome. Okay? Okay. So now, neurons have these different levels, meaning you know how I had that arrow going to another arrow? So each one is a different level. Neurons at these different levels are going to have different receptive fields. Okay? So there is this idea there of what's called the spatial frequency model, which says that the visual system analyzes the number of light, dark, or color cycles per visual space. So are you familiar with the idea of Hertz, which is how many cycles per second? Okay, that's not a good analogy. All right, so it's basically the number of how many light-dark cycles for a certain space in the visual field. And the reason is cortical cells, so in the cortex, are really best at detecting or sensitive to light-dark cycles or changes. So the cortical cells we talked about, for example, the simple cells, the complex cells, because now we're in the cortex, are going to be maximally turned on by bars of light, by lines, basically. So it's another way of saying what we said a slide ago about the complex and the simple cells. So the best stimulus to turn on these cells are going to be bars of light. That's what I mean by line. Okay, kind of like this. And in fact, in perception labs or some sort of visual lab, the material that's used to study about the visual system, a lot of the time they use very simple checkerboard or moving lines. Okay? Since you really wanted to know what happens beyond V1, I'm going to give that to you. Okay, so from area V1, these axons of these cells in V1 are going to then talk to the next level, which is V2. So you can now have another arrow going from V1 to V2. Okay. And then things from there can then bifurcate and go to different areas in the brain. So it can go to V4. We'll talk about what that is. And then it also can go to what's called the inferior temporal lobe, which is involved in seeing form. So at this point, you're actually then talking about shapes of things. So much more complicated than just lines. Now again, we're still each of the neurons, even in V4 and in the inferior temporal cortex, each of them are still going to have receptive fields, but these receptive fields now are going to be tremendously more complex than just a simple moving line or simple orientation. Now you're actually talking about shapes. Okay? V1 and V2 have very similar properties, but beyond that, things get very different. Okay? V2 adds, the complexity V2 adds to all of this, to V1, is that it responds also to illusions or illusory boundaries that actually don't exist. So this is just look at how big the visual area is in the back of the brain. That's pretty big. Okay. So here are some of the main visual areas, both in the monkey and humans. You have V1 at the very, very back. If your V1, for whatever reason, let's say you get shot, like in the very back here, you are cortically blind. Even if your retina is completely fine, you are blind. Okay? And then you have V2, and then beyond V2, things get a little bit complicated with each of these different colors processing or having different visual receptive fields that add different information. Okay? Here's the inferior temporal area in, I think that's what, a plum color, I think, I don't know. So here, or purple, this is going to process the shapes that we talked about earlier. Okay. And V5, the medial temporal, also we're going to talk about it because it's going to be important for movements. Okay. And then, as I said earlier in the last lecture, don't forget a small branch. Most of it goes LGN to V1. But don't forget that also a small branch of the ganglionic axons actually go to the what? The what? In the midbrain? What? Yes, you said it was the person next. Superior colloquius. Okay. Okay, it's on there. Okay. So, and again, the fovea is going to have a big area there in the cortex. But this V4, can go up to, I've seen up to 30. So, but V1, V2, V4, and then we're going to talk about the inferior temporal and movement. So, you see that triangle that's there in the middle, that's actually, there's no line? Your eyes actually feel the same. In reality, there is no triangle there. But your eyes continues this or completes it as though there's a triangle there, even though there isn't.
That's illusory.
Yeah. Yeah. Okay. Your eyes does tons of these where it can, you have something. So here, for example, you have also a discontinuation in the triangle that, with the actual line. But really, there is no triangles. Your eye just makes that happen. And it's very difficult to prevent this perception, oh, this is a triangle. It's very difficult to do that. It's very difficult to override what your eye or what your brain has already perceived this as. Okay. Some areas in V4 cells have very strong responses to concentric and what's called radial stimuli and to color. So V4, a lot of people think about V4 as very important to your perception of color. The inferior temporal visual cortex is going to respond to, or its receptive fields are going to respond to very complex shapes, which sometimes then you can have the information from V4 adding color, and then you can actually have texture added to that as well. The inferior temporal cortex can actually be trained through your experience and learning. So it's receptive fields and what kind of shapes they can see or perceive that actually comes through your regular experience in your environment. Yes?
Wasn't there an experiment, like they raised a cat in a box where it could only see
Horizontal lines?
horizontal lines, and then when they let it out in the world, it couldn't see
Any other orientation, yes. And they did this to dogs too. They have them with cones where you can only see one orientation of a line. They can't see any other orientations. And then they're basically, when they take it out after the critical period, they fail to see any of the other lines.
So that's because their brains didn't develop.
Exactly. So the hardware is there, but if you don't train it with proper experience, I'm not saying you go and buy elaborate toys that are us, things or babies are us or whatever it is. I'm saying that you provide your infant with lines, corners, very simple things. If you've ever been around toddlers, you know that they will leave an expensive toy and play with a Kleenex box. But you find the toy interesting, though. I can never assemble these things anyway. Okay. And in fact, in the inferior temporal cortex, there is an area there that's called the fusiform gyrus, and sometimes it's called the face area, because some believe that it's highly specialized area, specialized for just seeing faces or distinguishing among faces. Think about it. We all have two eyes, we all have one nose and a mouth, and we all have the same, but yet we are so different. And our brain has a very, very good ability at distinguishing among the faces. Okay? You see in the sky the two holes and a line, you think you're seeing a face talking to you at night. Okay? Yes, we all do that. Okay? And that stems from the fact that your brain has a module in there that is so specialized for seeing faces. Yeah.
What was that for?
The fusiform gyrus, and sometimes it's called the face area. And the prefrontal cortex is also neurons that are going to respond to faces as well. Now, so we talked about the inferior temporal cortex in terms of seeing shapes, or kind of like what information. Okay. You also have another branch that's going to parallel to the inferior temporal, going to V5, which is called the medial temporal area, that responds to movements. Okay. So from V1, and then it goes to V2, and then to V4, and then you have information kind of going to the inferior temporal, and parallel to that, you have information going to MT or medial temporal. So you have the what information and where information, two parallel pathways. Okay, so now,
Just to clarify on the pathway, it goes from V1 to V2, and then it can either go to V4 or the inferior.
no, and then it can go to inferior temporal or the medial temporal, which is also known as V5.
So where does V4 place?
So V1 to V2 to V4. Okay, but you can have it go to V4, or you can just skip the V4 for now. And then you have the bifurcation where you have two parallel pathways going to the what information or the inferior temporal, and the where information, which is MT medial temporal or the V5. You should know that they're both synonymous. So V4, here are the responding to color and texture. The inferior temporal, you now are starting to get much more complex receptive fields. You see that? Much more complex receptive fields. Again, this is something the receptive fields of these neurons is going to be attuned to whatever environment you're in. Okay? Okay. Color is kind of created by your visual system, but really there is no such phenomena out there as color. What we talk about is the wavelength and the wavelength and how many nanometers. That's what we talk about, but there's no such thing as actually colors. Okay? So we talk about the wave, the movement of that wave of photons, and how actually the brain makes sense out of this. The difference in wavelength causes your brain to perceive this as color. So the reason why you see red as different than blue, because the wavelengths are different from red and blue. Okay? And that's why your experience, your subjective experience of red is different than my subjective experience of red. We can both say red, what we're seeing is red, but what we subjectively mean by it might be, the shade of it might be different. Different than saying this is a chair, the chair is actually there. Does that make sense? So here you have the electromagnetic spectrum, which is basically all the different radiations there. You can only see what's visible to you. So all of these are wavelengths. Okay, this is the wavelength there. What you can see out of all these different radiations, very, very narrow bands of visible light that you experience as color. Okay? So blue, or maybe violet here, is 400 nanometers, while red is 700 nanometers. The reason why you're seeing red is because that your brain is interpreting 700 nanometers to correspond to your perception of red. Okay. Yes.
I just had a quick question because I read somewhere online and I kind of don't believe it, but somebody said that
You don't believe everything that's written online?
I didn't believe hardly anything that's online, but somebody said that there's no such color as pink, like in wavelength, and it's just that what we perceive as pink is that our brain doesn't know what to do with that color, so it puts pink there, but there's no like wavelength of light that corresponds to pink. Is that?
No, you can
BS?
measure the wavelength of anything. Okay, visible or not visible. Okay? Okay, so some of your microwave, these are waves. These are also waves, but they're not visible to you. Radio waves are not visible to you. X-rays are not visible to you, although they exist. That's why they ask you, are you pregnant before getting X-rays? Okay? So all of these waves exist, but you can only see parts of it. Okay? Does that make sense? So this is the solar energy when it hits Earth, traveling through a wave. Some of it we can perceive, some of it we can't. Your radio, like if you're listening to NPR this morning, 89.5 or 43.9, depending on where you live, that's also a wave. Okay? You can't see that, but you can actually use it to make sense of it. Like, you know how a few years ago, the government basically wanted us to replace all our analog TVs with just like the digital ones? That's because now we've got too crowded in terms of waves in the sky. Because that's what it was using. Remember the antenna? Maybe you're too young to remember this. And fixing the antenna and all that. Okay? You can tell your kids that about that. Okay. So an object's color is going to depend on which wavelengths are reflected. And you can actually have something to measure that. Okay? So the other colors are going to be actually absorbed. So there's going to be 3 dimensions to color perception. There's what's called the brightness, which is going to be from dark to light. There's what's called hue, or what you probably know as just color. Yellow versus blue. Okay? And then saturation, which is how pigmented, how full that color is. These are 3 different ways to describe your perception of color. So short wavelengths, does that make sense to you? Do you know what does short wavelength mean? What's a wavelength?
There's very little space between the peaks.
Exactly. So when we talk about the wave, it travels like this, right? There's peaks. The distance between 2 repeating peaks, that's a wavelength. When that difference is short, this is what I mean by short wavelengths. That corresponds to what you see, kind of like the bluish, purple, violets. These are short wavelengths, which also means that you probably have too many peaks, so higher energy. Okay. A long wavelength means that the distance between the peaks is larger. And it's probably a slower wavelength, okay, with lower energy. So if you remember, the visible spectrum was something like 400 nanometers, kind of the blue-violet, all the way to 700 nanometers. So the distance between the peaks. So this looks blue because it takes in, it absorbs, or subtracts most of the long wavelengths and some of the medium wavelengths. So what is reflected is only the short. Okay. Does anyone here do stage lighting? No. Okay. All right. I'm going to use you. Okay, never mind. All right. So we're not, when you're thinking about this, don't think about mixing colors like, you know, on a paper. Because what happens when you keep adding colors? It becomes the brown or black. It's different. This is light. Here, what we're talking about is mixing light. Different. So the reason why this looks blue, which is the short wavelength, is because the medium wavelength is being absorbed. The long wavelength is being absorbed, which means what's being reflected is the blue. Or sorry, the short wavelength, which corresponds to psychologically what we see as blue. The reason why that in the middle looks yellow, which corresponds to somewhat like a medium wavelength, that's because what is absorbed here is the short and some of the long, so you get what's reflected back, the middle wavelength. Okay. Now, what happens if you mix blue and yellow? If you mix these two together, then what's getting reflected are these two, and what's getting absorbed is the other one. Okay. So that's how we actually see color. Okay. So you have short wavelength, medium wavelength, long wavelength. The short wavelength kind of corresponds to what we know or experience psychologically as the blues. Not as your sand, but as the color blue. And then the medium wavelength, you have there what? The green, yellow. The long wavelength is you have the red. So if what's being absorbed is the short and medium wavelength, then what's being reflected are the long wavelengths, and you experience red, and so forth. Now, white light contains all colors, all visible colors. So if you, and you might have done this in science lab, if you shine white light through a prism, what do you get coming out of it? The rainbow. It separates all the different colors that make it up, which it contains all colors. Okay? So here on top you have white light, and then you have the brightness from top all the way to the bottom, from light all the way to black, from light to very dark. This is all the different hues there going around. Saturation has to do with how really pigmented that color is. So when you're doing a PowerPoint, sometimes you manipulate this. So the color could be very pigmented to not as pigmented. Okay. Around is the hue or the different colors, right? Going blue, red, yellow, or green, and so forth. And then going this way, this arrow is the saturation. And then going from white to black is the brightness. Okay. The two middle squares, if you cover all the surroundings in both of them, they're actually the same exact color. You can try it at home. All right. So color perception is going to also involve some of the receptive cells, and it's going to take advantage of their different sensitivities. So remember, we talked about different types of photoreceptors that have different types of photopigments. Okay? If you remember from, not last lecture, but I think the lecture before that, we talked about 3 different types of phones with 3 different types of oxygens, 3 different types of pigments. Remember that? And remember we talked about rod with its pigment rhodopsin. That's going to be very important for this. There's what's called the tri, meaning 3, chromatic. Chrome, does anybody know what chroma means? Color. The trichromatic hypothesis of how we see color. There's different ideas about how we actually see color. The idea here, or the hypothesis, according to this hypothesis, is the reason why we perceive color is that because we have 3 different cones with 3 different options, 3 different sensitivities to light. That allows us to see different colors. Each of the cones is going to respond to a different part of that electromagnetic spectrum that I showed you, particularly the visible part. And each of these cones, the 3 different ones, is going to have its own dedicated pathway to the brain to alert it that this is a different color. So then color is going to be recognized, or your perception of color is going to happen depending on which receptors are activated, which cones are activated, which photoreceptors are activated. Okay? And some 100 years later, somebody else came up with another theory of color and how we perceive color that actually it's called the opponent process hypothesis of color. And what that is that we have 4 unique hues and 4 opposite pairs. So blue is the opposite of yellow, green is the opposite of red, and then some say black and white. So we used to think, well, it's either one or the other, which theory is correct in terms of seeing perceiving color. And as it turns out, they're actually both correct. But they're both correct at different levels of that visual pathway. Did I lose you guys? Did I lose you guys? No? Yes.
So they're both correct in certain points, right?
So remember how we talked about different layers of complexity in the brain, going from the bipolar, or going from the retina, to then the LGN, to then the cortex? That's what I mean by levels of complexity. So both these theories are correct, but there are different stages of processing. Okay? So the trichromatic is correct at one of these levels, but then the opponent process is correct at another level. So both are working. Okay? So for the trichromatic hypothesis, it really makes sense if you look at the cones or the photoreceptor. That hypothesis really makes a lot of sense. Because you have 3 cones that are going to respond or are going to have peak sensitivity to the short wavelength, medium wavelength, and long wavelength. So here you have the cones by themselves are not color detectors. It's the relative activity of these 3 cones that tells you about the color. So I don't want to make it sound like activation of this, the middle cone, you'll always see green. It's what's going on in the 3 different cones, their average activations, that tells you what color you're seeing. Okay? So for example, okay, and you see the rods are also there. So the rod, or your night vision, is much better at 495 nanometers, which is about, the bluish, going a little bit into the green. So that's your best perception will happen to colors that correspond to that at night. Oh, wait a minute, that also happens to be the same color as the sky at nighttime. Makes sense. Okay? So red is very difficult to see at nighttime. It actually appears kind of blackish at nighttime. Why don't we start with this next time, because I'm just getting my internal compass is saying I've lost you 5 minutes ago. So let's start with this. Now, next time, we were supposed to, I don't know if you've forgotten or you think I've forgotten, so you don't want to remind me. We were supposed to have an exam on Monday. Do you remember that?
Yeah.
Genius people. Okay, so what we're going to have the exam on Wednesday instead. But, no, don't take a breath. But we're going to have a quiz, surprise quiz potentially, on Monday. Okay? And what we've covered so far on the visual system. This will be hopefully a wake-up call, okay, for the quizzes. Some of you, not all of you, you did abysmal on the last quiz. So make sure, we've slowed down a lot for this chapter. You have the PowerPoint. Make sure you study it really well so you can be in good shape, because you won't have enough time to study on Tuesday for the test on Wednesday. There is no way. Okay? So on Monday, we're going to do a check-in with a quiz, potentially, on what we've covered so far in the visual system, and we've covered quite a bit. We'll do the quiz, we'll finish this chapter, see if you have any questions for that, and then we'll have the exam. All right? See you guys next time.